Welcome to today's video. Today we will discuss the post-translational process of proteins in cells. After a protein is synthesized inside the ribosome, it undergoes several stages of modification and targeting to ensure it functions correctly. We will explore these stages together step by step. Part 1. Protein folding. The first step after translation is the process of protein folding. When a protein is synthesized inside the ribosome, the amino acid chain is a long and non-functional line. Therefore, this protein must fold to take its three-dimensional shape. This folding depends on the chemical interactions between the amino acids present in the protein. If the protein is not folded correctly, it may become non-functional or even toxic to the cells. Additional explanation: folding occurs thanks to helper molecules called chaperones that assist in ensuring the protein folds correctly. Part 2. Post-translational modifications. The next stage is post-translational modifications *, which are chemical modifications that occur to proteins after they are. These modifications are crucial in determining the protein's function and location within the cell. For example, phosphate groups may be added to the protein in a process called phosphorylation *, which helps to activate or inhibit the protein's functional activity. Common modifications include phosphorylation *, adding a phosphate group to the protein to activate or disable its functions. Glycosylation *, adding sugar to the protein, which may help it interact with other cells. Acetylation *, adding an acetyl group to control the protein's function. Additional clarification: post-translational modifications are considered extremely important because they alter how proteins operate within the cell. Part 3. Transport and targeting. The next stage is protein transport to the appropriate location inside or outside the cell. Some proteins function within the cell in the cytoplasm, while others must be transported to organelles such as mitochondria or nucleus. Proteins that need to be transported outside the cell include proteins secreted from cells such as hormones or digestive enzymes. This occurs through a complex network of cellular tubes called the endoplasmic reticulum *, where the protein is modified and packaged, then sent to the Golgi apparatus. From there, the proteins are packaged into vesicles and sent to their destination. Part 4. Protein function. After targeting, once the protein is directed to its correct location, it begins to perform its function. Proteins can perform many functions in the cell, such as enzymes that accelerate chemical reactions, receptors that receive signals from the outside, structural proteins that provide support to the cell. These functions depend entirely on the protein's structure and folding, as well as on the modifications it underwent after translation. Part 5. Protein degradation. And finally, when proteins are no longer needed or become damaged, they are broken down by various mechanisms. In cells, lysosomes and proteasomes * are used to destroy proteins that are no longer needed. Clarification: the proteasome is a complex that recognizes damaged or unused proteins and breaks them down into their basic components for reuse. Thus, proteins are not simply made in the cell. They undergo several complex stages before they can perform their tasks properly. From folding, to modifications, to transport, then performing functions, and finally degradation. All these processes are important to ensure the integrity and efficiency of cells. If you liked the video, don't forget to subscribe to the channel for more useful scientific explanations.